Cytoplasmic determinants are maternal substances located in the cytoplasm of the cell. These consist of messenger RNA, proteins, organelles, and other substances. Because they are distributed unevenly throughout the cytoplasm, this affects the early development of the embryo. Through mitosis, the two cells are the same genetically, but because of the cytoplasmic determinants, the genes expressed will be different. As embryonic cells divide, cells must communicate with each other for proper gene expression. This is done through a process called induction. Cells are greatly influenced by their environment, just as much as what is inside the cell itself. Induction is a major source of developmental information. Neighboring embryonic cells secrete signals such as growth factors, which affect the development of the target cell. It causes changes in the gene expression and ultimately changes in the cell itself.
All cells start out as embryonic precursor cells. These cells are pluripotent, meaning that they can transform into multiple types of cells. Some examples include muscle cells, others are white blood cells, and some can even transform into neurons. All the following forms of cells are unipotent, meaning that they can only stay the type of cell they already are throughout their reproduction. For the following example, we'll be using a muscle cell to show how transcription plays a factor in the process of cell determination. All cells start out as embryonic precursor cells, and here in this example we have a muscle cell following it, and then a muscle fiber afterwards. In the DNA, we have master regulatory genes and muscle genes. Both are usually off in the embryonic cell, but when the muscle cell finally forms, the master regulatory gene is transcribed to form a transcription factor that activates other muscle-specific genes for transcription. This, in turn, creates other muscle-specific proteins that creates the muscle fiber that we see.
Apoptosis is programmed cell death. In this process, DNA, organelles, and other cytoplasmic components are chopped up, packaged into vesicles, and engulfed by scavenger cells, leaving no trace. This process is crucial in developing embryos, and it is the reason why there are spaces between our fingers.
In the embryo, we also have pattern formation, which determines body structure and orientation. Along with pattern formation, we also have positional information, which are molecular cues such as cytoplasmic determinants and inductive signals that control pattern formation. Here we have a stretch of DNA with a homeotic gene expressed on it, which makes sure that all body parts are organized in the correct order. A mutation in the homeotic gene can cause a body part to grow in an incorrect place, and a misformity severe enough to kill the embryo is called an embryonic lethal. In the embryo, we also have the issue of axis establishment. For the purposes of this example, we'll be only talking about anterior and posterior orientation. A maternal effect gene is a mutation in the mother's gene that will create mutant offspring, regardless of the offspring's genotype. One example of this would be an egg polarity gene, which determines anterior and posterior axis formation. One example is the bicoid gene in flies, or the Drosophila. Here we have two eggs, one with a normal distribution of cytoplasmic determinants for the bicoid gene, and another mutant cell with abnormal distribution of the same cytoplasmic determinant. The different gradients are called morphogen gradients, and an abnormal gradient in the mutant cell causes a fly to be produced with two tails instead of a head and a tail. That concludes our presentation. Thank you for watching.